A hydrogenosome is a membrane-enclosed organelle of some anaerobic ciliates, trichomonads, fungi, and animals. The hydrogenosomes of trichomonads produce molecular hydrogen, acetate, carbon dioxide and ATP by the combined actions of pyruvate, ferredoxin, oxido-reductase, hydrogenase, acetate, succinate, transferase and succinate, thiokinase, superoxide, dismutase, malate, dehydrogenase, ferredoxin, adenylate, kinase and NADH, ferredoxin, oxido-reductase are also localized in the hydrogenosome. It is nearly universally accepted that hydrogenosomes evolved from mitochondria. In 2010, scientists reported their discovery of the first known anaerobic metazoans with hydrogenosome-like organelles. Hydrogenosomes were isolated, purified, biochemically characterized and named in the early 1970s by D.G. Lindmark and M. 1/4 at Rockefeller University. In addition to the seminal study on hydrogenosomes, they also demonstrated, for the first time, the presence of pyruvate, ferredoxin, oxido-reductase and hydrogenase in eukaryotes. Further studies were subsequently conducted on the biochemical cytology and subcellular organization of anaerobic protozoan parasites. Using information obtained from hydrogenosome and biochemical cytology studies, these researchers determined the mode of action of metronidazole in 1976. Metronidazole is today recognized as the gold standard chemotherapeutic agent for the treatment of anaerobic infections caused by prokaryotes and eukaryotes. Metronidazole is taken up by diffusion. Once taken up by anaerobes, it is non-enzymatically reduced by reduced ferredoxin, which is produced by the action of pyruvate, ferredoxin, oxido-reductase. This reduction creates products toxic to the anaerobic cell and allows for selective accumulation of the drug in anaerobes. Hydrogenosomes are approximately 1 micrometer in diameter, but under stress conditions can reach up to 2 micrometer, and are so called because they produce molecular hydrogen. Like mitochondria, they are bound by distinct double membranes, and one has an inner membrane with some cristae-like projections. Hydrogenosomes have evolved from mitochondria by loss of aerobiosis-related features in several lineages. In most cases, hydrogenosomes are genomeless, though genomes have persisted in some lineages such as Neocalymastics, Trichomonas virginalis or Trichomonas fetus. However, a hydrogenosomal genome has been detected in the cockroach ciliate Nyctothris ovalis and the Stramenophile blastocystis. The similarity between Nyctothris and blastocystis, which are only distantly related, is believed to be the result of convergent evolution, and calls into question whether there is a clear-cut distinction between mitochondria, hydrogenosomes, and mitosomes. The best studied hydrogenosomes are those of the sexually transmitted parasites Trichomonas virginalis and Trichomonas fetus, and those from rumen chytrids such as Neocalymastics. The anaerobic ciliated protozoan Nyctothris ovalis, found in the hinget of several species of cockroach, has numerous hydrogenosomes that are intimately associated with endosymbiotic methane-producing archaea, the latter using the hydrogen produced by the hydrogenosomes. The matrix of N ovalis hydrogenosomes contains ribosome-like particles of the same size as a numerous type of ribosome of the endosymbiotic methanogenic archaea. This suggested the presence of an organelle genome, which was indeed discovered by Ekman-Over and later partly sequenced by Boxmer. 3 multicellular species of Loriciferus Piniloricus nov. sp. Rugiloricus nov. sp. and Placiloricus nov. sp. have been found deep in Mediterranean sediments and use hydrogenosomes in their anaerobic metabolism cycle.